Am I audible? Hello?
Yes, you are audible.
Yes.
Carry on, sir.
So lovely evening and heartiest welcome to all of you in the fifth day of the online lecture series for undergraduate studies, which got its onset on 25th of May 2024. In this very moment, we have with us Dr. Ujjwal Malik, who happens to deliver his lecture on the topic vaccine, which is scheduled in semester 4 in the paper 10, that is immunology. Now I owe the opportunity to request Dr. Rajnaran Roy to formally acquaint Dr. Ujjwal Malik to all of you.
Thank you, Dr. Mojumdar. It's my immense pleasure to introduce Dr. Ujjwal Malik, today's speaker of the ongoing summer lecture series of undergraduate student zoology organized by Zoological Association of Burdwan. Dr. Malik, presently associated with Netaji Mahavidyalaya as an assistant professor, Department of Zoology. He is associated with this post since January 2017. He has completed his MSc from Vishwabharat University, Santiniketan, with environmental biology as special paper. His research interest includes the following field: spider sexual behavior, tritrophic interactions, ecology, plant-insect interactions, antioxidant enzyme response, nutritional ecology, and insect. He has published 14 research articles in peer-reviewed journal having average impact factor 1, total citations 1.49, H index 7, I10 index 7. He has presented his deliberations at 6 conferences. He has received a large number of awards in his academic career. He was awarded GATE conducted by IIT, Ministry of Human Resource Development, Government of India in 2011. He have qualified MTech PhD admission program 2011 at IIT Kharagpur. He have qualified SET conducted by West Bengal College Service Commission in December 2014. He was awarded as a Junior Research Fellowship 2012 and Senior Research Fellowship 2016. He has a broad award. He was awarded postdoctoral researcher, Jingjiang Sun University, China, 2019, and awarded short-term researcher award, Jiangsu University, China, 2023. So I would like to invite Dr. Malik for his deliberation. Dr. Malik, please.
Okay, thank you so much to invite me. Anybody else who do not understand Bengali? Sarojda? আছে? Okay. Is it visible, my slide? Is it visible, my slide? Please response.
Yes, visible.
Yes, sir.
Okay. So today we are talking about vaccine. Already I have been introduced by Dr. Rajendra Rai. So thank you so much for again inviting me. So before to understand the vaccine, some few terminology you have to know. First one is the immunity. So basically, what is immunity? What do you understand immunity? That means it's protection from any infectious disease. Last COVID-19 has been happened last 2 years ago. You know very well, kind of that. So if you are immune to a disease, you can be exposed to it without becoming infected. So for the immunization, one of the most important thing is that vaccine. So that's why during the COVID-19, every rich country and poor country all are trying to make the vaccine. So who prepare first? They are people, the country people. They have been saved by the vaccine. So in our country, in India, there is two most protectable vaccine was there that time, Covishield and Covaxin. A lot of people saved their life by this vaccination. So by this page, you really understand what is the importance of vaccination. So basically, the term is vaccine means a preparation that is used to stimulate the body immune response against disease. Say, for example, COVID-19 like that. So first round when it has been started 2020. I think that is the month of March. First COVID came China, then whole world it has been expressed. So first vaccine I think is in 2021 like that. So after that, next one or next 3 or 4 phase of COVID-19 that has been immunized by the vaccination. So vaccine are usually administered through needle injection. We know very well, or it can be administered by mouth or sprayed into the nose. That depending on the vaccine, it's different theory to inject the people or to take up the people. So what is vaccination? The act of introducing a vaccine into the body to produce protection from a specific disease. Say for example, against the COVID-19, Covishield and Covaxin, a lot of vaccination are there. So by the process, an individual or a person becomes protected against a disease through vaccination. So that is called immunization or vaccination. This term is really almost same, vaccination or immunization. So if we look at the history of vaccination, that it has been started 7th century, oldest Indian history, that Indian Buddhists drank snake venom to protect against snake bite. I think maybe you know the story. Then 10th century, variolation to prevent smallpox in China and Turkey. Early 1700, again in England, it has been happened variolation. And the Generian era, that is 1760 to 70. So that is that time, the Generian era, it has been started, and then 1875 to 1910, dawn of immunological science. Again, toxin and toxoid, early bacterial vaccine, it has been happening in 1910 to 30. So after that, influenza, yellow fever and influenza came into the world vaccination, early viral vaccine 1930 to 50. Then again, mumps, measles, poliomyelitis, lot of vaccination came. So finally, glycoconjugate vaccine and rotavirus vaccine, that is modern vaccine, human papillomavirus vaccine and herpes zoster vaccine. So a lot of vaccination today is invented by the scientists, and still now a lot of vaccination process is going on. If you see the vaccination history or vaccination center, a lot of vaccination is still going on. So Edward Jenner, that the greatest scientist, he published the test immunity by challenge with smallpox. Then the Louis Pasteur, very well invented rabies, and Robert Koch, very popular scientist, killed whole cell bacteria vaccine, that is Vibrio cholerae. So what is the basic concept of vaccination? Why we do concentrate about the vaccination? So because it protect those at a highest risk. That means selective immunization or strategy, or to eradicate, eliminate, or control disease. So before polio, say for example, before polio vaccine, the story, lot of infants have been died or have been infected by polio. But right now, polio is under control. Very, very less baby are infected by polio, because this is the great success of vaccination, polio vaccine. So currently, it is estimated that vaccination saved the life of 3 million children a year. Can you imagine that? So eradication, that means infection, any pathogen has been removed worldwide, smallpox, rinderpest, eradication. Elimination, what I am talking about, polio, measles, disease that has dispersed from one area but remains somewhere, it still remains. So, and finally control. So disease no longer constitute a significant public health problem, that is neonatal tetanus. So basically, before understand the vaccine or vaccination, you need to understand the two immunization. One is called passive immunization, another is called active immunization. So see this picture. So what is the passive immunity? That is natural immunity, that antibody like that we usually get from our mother, that is maternal antibody. Another thing is that artificial antibody, monoclonal antibody. If you get this antibody, that is also passive immunity. So what is active? So one of the most important active immunity is that artificial or vaccination, either or you get infected one of the any natural pathogen. Suppose বাংলা একটা কথা আছে, খুব প্রচলিত কথা, যে একবার যদি কারো পক্স হয়ে থাকে, সো সেকেন্ড টাইম পক্স হওয়ার চান্স কম থাকে। সেই কথাটা আমরা সবাই দারুণভাবে শুনে এসেছি ছোট থেকে। সো দ্যাট মিন ইজ দ্যাট you get the natural immunity from that particular pathogen. So this is also a active immunity. So suppose you got a vaccine, you have been got a vaccine, any vaccine. So this vaccine, suppose you take a vaccine 1 month ago, 2 months ago, or 1 year ago. So after 1 year ago, after 1 year, after later 1 month or 2 months, 3 months, so vaccination and vaccine, that what have been produced the antibody like that, so that the is still written in the memory cell. So this is called active immunization. So both stimulate the proliferation of T cell and B cell. I think you know the T cell and B cell, that resulting in the formation of the memory cell. So the formation of the memory cell is the basics for the relatively permanent vaccination. So for example, you know the baby, when baby birth, that is within 24 hours, the vaccine that is called BCG have been put. So one time BCG have been put. That means it retains the baby body in the case of memory cell, look like memory cell. This is the one example of memory cell. So if we compare the serum antibody and immunologic memory, so suppose this day antibody or vaccine has been put here, and 1 year or 12 months, you see the immunologic memory that has been rise, but serum antibody in your blood, that will drop down to down. So if we compare to these things, so you got the immunologic memory that is permanent, almost that is permanent. So again, if you infected particular pathogen or disease, that memory shell try to protect your body. So immediately they can act, they can work against your pathogen or against your antigen. So that's why memory cell is very much important in case of vaccination. So what is the property of a good vaccine? So it has to be ability to elicit the appropriate immune response for the particular pathogen, or it has to be long-term protection. The second thing is the most important, long-term protection. Otherwise the vaccine will be cost benefit and safety, and it has to be also stability. And of course, this is the most important thing, like our country, India. So this is inexpensiveness. That is most important thing, inexpensiveness. That the thing. That's why COVID-19 vaccine, our government of India freely they donated this vaccine. So if we see the type of vaccine, there are so many types: live vaccine, killed or whole organism vaccine, subunit vaccine, purified or recombinant antigen, or DNA vaccine or recombinant vaccine. So what is the live vaccine? Say for example, typhoid vaccine. So these vaccines are prepared from attenuated strain that are almost completely devoid of pathogenicity. So what is the mean of that attenuated? Suppose we can make a vaccine from any pathogen. So first you need to modulate the pathogenicity. Any pathogen like X, that has to be modulated, that has to be attenuated. That means it has been modulated, the pathogenicity, but are capable of inducing a protective immune response to the body. So they multiply human host and provide continuous antigenic stimulation over a period of time. Second is that kill or whole organism vaccine. That means this is the most example of the covaccine. It is a vaccine that is produced by growing the organism and then killing or inactivating. So in every vaccine, in case of whole organism or in case of live vaccine, the pathogenicity has to be inactivated or it has to be modified either by heat or by chemicals. So in case of whole organism vaccine, it has to be modified by the heat or chemicals. So that's why these are used when safe live vaccine are not available. So that time we can use the kill whole organism when the live vaccine is not available. So for example, inactivated polio vaccine or rabies vaccine. Third one is the subunit vaccine. Subunit means subunits are defined as those vaccine that containing one or more pure, semi-pure antigen. Either it may be pure or semi-pure. So these are the three types: toxoid, recombinant subunit vaccine, and non-recombinant subunit vaccine. What is toxoid vaccine? Like diphtheria and tetanus. So it is not the growth of the bacteria, but the dangerous thing is that the protein toxin that is liberated by it, diphtheria. So treating the toxin with formaldehyde, that is chemically modulated formaldehyde, that denatures the protein so that it is no longer dangerous. So you cannot put that toxin directly to the human body or any animal. So you need to be modified either by chemical or by heating. So here it has been modified by the formulated. That protein has been denatured by formaldehyde, and that's why the pathogenicity or dangerousness has been removed. So that's why in this way, in this toxoid vaccine has been inactivated. So it's called inactivated toxin is called toxoid. So for example, DPT vaccine also called triple vaccine. Next is the subunit recombinant vaccine. So these vaccines are those in which genes for desired antigens that are inserted into a vector, usually a virus that is in very low virulence. So I will show the this one. So this is the DNA vaccine. Just a second. Sorry for disturbing. So Saroj, just 1 minute. Just a second. দেখা যাচ্ছে তো? আচ্ছা ওকে ওকে আমি ফার্স্ট নিয়ে নিই। আচ্ছা, that is DNA vaccine. What is recombinant? One type of disease recombinant vaccine. So this is the plasmid that if you interested a particular gene, so you can input in the plasmid. Say for example, this is the gene that is for antigenic protein. You need to interest this type of gene. You can put the DNA vaccine, particular gene. So when you can input the plasmid, it can incorporate it into the individual, so that it produce, it's a lots of protein. You know the central dogma. By the central dogma, it produces the protein. So that protein in your body, that is antigenic peptides. So this antigenic peptide will react with your T cell and B cell. So T cell, here two type of T cell, you know, TC and TH. TC means hydroxy T cell and TH T helper T cell. So that's why two type of T cell has been activated. And in case of B cell, in case of B cell, it produces a lots of antibody. One type of antibody it has been produced, and that antibody will memory in your body. So particular gene is injected here, and finally you got the two type of cell mediated and humoral response. One is the antibody mediated, another is the T cell mediated, and you got the lot of immunity from this type of vaccine. So I'm talking about this way. This is the subunit recombinant vaccine. So these vaccines are those in which genes for desired antigens are inserted into a vector, usually virus, that has been very low virulence. But this vector expressing antigen may be used, vaccine or the antigen may be purified and injected a subunit vaccine. So the only recombinant vaccine currently in use in humans is the hepatitis B. Or maybe this is the much safer than using attenuated HBV, or which could cause lethal hepatitis or liver cancer if it reverted to the virulent phenotype. So recombinant DNA technique can also be used to make safer attenuated pathogen vaccine. So second is that which vector to be used. That is the most important thing to continue this type of vaccine. Must be compatible with host cell system, that is prokaryote vector for prokaryotic cell or eukaryote vector for eukaryotic cell. So need a good combination, strong promoters, ribosome binding site, termination sequence, affinity tag or solubilization of sequence, and multi-enzyme restriction site. So this is the most important thing, which vector you can use in this type of vaccine. So recombinant virus vaccine or live recombinant vaccine, here you know already I have discussed a particular gene that code for immunogenic protein from one organism into genome of the other, such as vaccine virus is introduced. So the organism expressing that gene is called recombinant. I already explained the picture in case of DNA vaccine. So following injection into the subject or human individual, the recombinant will replicate and express sufficient amount of foreign proteins that to induce a specific immune response to the protein. That means humoral or cell mediated immune response. So what are the advantage of viral vector vaccine? Some advantages are there, that it elicits strong humoral and cell mediated immune response that resulting in memory. So that's why this is the most important advantage. This type of vaccine, like recombinant or DNA vaccine, so it can be targeted by viral tropism for particular cell, or like example intestine, brain, that inducing desired immunity, or it can also encode for several antigens from different pathogens, introducing the possibility of a single vaccine for several diseases. So viral vector have been found not to interfere with the protection produced by the other type of vaccine. So vaccine are relatively inexpensive or some easily transportable. That is, has to be cheap and has to be easily transportable. Suppose in a rural area, so when the center from the hospital to the RCDC center like that, when someone carry the vaccine, so you cannot control the temperature, which temperature actually they need to preserve, but sometimes it has to be changes. So that tolerance has to be accepted, otherwise it can be destroyed. So that's why it has to be easily transportable. That is most important thing, is transportable. What are the disadvantages? Since the live is being used as an attenuated form of human pathogen, so there is always a risk of reverse virulence. Maybe sometimes it has been happened. Some vectors under, like adenovirus, have the capability of transferring cell to cancerous phenotype. So that's why whilst oncogenes are removed, virus could, virus could recombine with natural occurring pathogenic strain in the environment and new hybrid virus transfer new properties. So immunoresponse to virus infected cell may cause pathological problem. The disadvantage. So one kind of another recombinant attenuated, same thing had been happened, what I've already explained. So comparison, that is vaccine and recombinant, the discuss that city or virulence has been by the chemical or physical activation, laboratory live attenuated, isolate, relay, attenuated, and recombinant or recombinant generated subunit DNA vaccine, vector-based organism to deliver foreign gene products. So what is the future development? So identification and utilization of beta immunogens as a new vaccine. Better vaccine delivery method, that is oral, intranasal, and system, allowing mass vaccination. So if the methodology is the oral, so a lot of people taking the vaccine, because some people or some individuals are very worried about the injection like that. So that's why oral is very common method. So use of immunomodulators in vector-based vaccine like CpG motifs or cytokines, expression of foreign protein in plants and development of edible vaccine. So vaccine developed for a non-infection agent, control and prevent cancer, or vaccine of induce long-lasting contraceptive. So this is the additional fact. Dr. David Stracher, who presented a more social rather than technical view of vaccine, that is, barriers to health care include 7 US, that means uninsured, underissued, underrepresented, uninspired, untrusting, ununiform, ethical variation. But some significant thing is that by this individual, $10 billion per year, 36,000 elderly die yearly from influenza despite the availability of vaccine. So social inputs is needed to lower these figures. It need to be low. So this is the most controversial thing like right now, that is Covishield are rare side effect in election season, dangerous of politicizing the vaccine. So you know the social media, I think lots of things have been happening like this. So someone talking about that is Covaxin is more better than Covishield in case of side effects. Someone talking about the no Covishield is better than that. So lots of people are defined, but by this situation, the people on social media and even some mainstream channel also are having their "we told you" so movement this week after the pharmaceutical company AstraZeneca admitted in court to the side effect of its COVID-19 vaccine that is manufactured and marketed in India as a Covishield. So this follows a story in the Telegraph and UK on April 28 about a class action suit brought against the company. So that is the report says AstraZeneca has admitted for the first time in the court they has to be uh, uh, got the document and they has to be admitted that the its COVID vaccine can cause a rare side effects. So what is the side effect is that thrombosis and thrombocytopenia syndrome, that is TTS, which translated to blood clots and low levels of platelets, a type of blood cell that prevent bleeding. So some people got this type of side effect, not all people. So very rare side effect who are already taking up the Covishield vaccine during the COVID-19. In case of Covaxin, there still now there is no report and there is no rumors like that. So I suggest you don't believe this type of rumors like that. And most important dangerous thing is that the vaccine fraud. So still, like our country or different country, vaccination fraud is still going on. So before taking the vaccine, you need to clear from where you can take it and what is the vaccine exactly, and you need to check also internet, also the company, a lot of things you need to check before taking the vaccine. So COVID-19 vaccine fraud, people are want to be alert to this scam. The NHS never asks for payment like that. That time it has been happened. So NHS alert the people, common people, and never asks for your bank details. Never arrive around at your home or administer the vaccine. Never asks you to prove your identity, the sending copy like that. So this is the also the take-home message. So I think you better know the look like fraud that like that. So I don't believe the any type of error like that, the rumor like that that has been already social media and WhatsApp group. It has been happened. But so to justify your knowledge, you need to understand what is actually going on. If something has happened regarding Covishield or Covaxin, you need to check their website. So their website always they always try to notify if something has happened like that. So before falling trap, you need to check and need to alert. So this is the take-home message, and thank you all uh, to participate here, this join this session, and thank you all. I am very much thankful to participate uh, this kind of lecture series. Thank you so much for this. This is all about the vaccine.
Many thanks to Dr. Ujjwal Malik. Now is the interactive session. So if
Yes,
you
yes,
have
yes.
any question, you can ask Dr. Malik. It's very important topic. So I think you have a lot of questions in this regard. So you can ask Dr. Malik. We have ample time in our hand. So you can ask any question. This is a very interesting topic, and I think so many questions are there, rather queries are there. So please, you go ahead. Oh, no question. I don't find any question in the chat box even. No questions. Uh, so, Joel, I think no questions are there.
Okay.
So I am very much thankful to you for your nice deliberation. Thanks a lot. I thank all the students present today